So imagine a massive, sprawling city and like a catastrophic earthquake hits.
Right. Just completely flattens the place.
Exactly. It reduces every single building to just this giant pile of rubble. And if you sort through that debris, you can count exactly how many bricks there are. You can count the glass windows, the steel beams. You basically know all the ingredients.
Yeah, you have the exact inventory.
You have absolutely no idea what the city actually looked like before the quake. I mean, you don't know where the hospitals were or like if the police stations were next to the banks.
Which is pretty useless for urban planning.
Exactly. And the crazy thing is, for decades, that is essentially how the medical field has been trying to understand and cure cancer.
It really is a sobering reality. I mean, when you look at traditional oncology, we've had to rely on breaking the tumor down, literally grinding it up to understand what's inside it.
We've been looking at the rubble.
Right. We've been looking at the rubble. And because of that, our diagnostic landscape has historically been, well, quite murky.
But that murkiness is clearing up, and that's why we're here today. Welcome to today's deep dive, where we are exploring just a massive, massive leap forward in precision medicine.
It's honestly one of the most exciting developments I've seen in a long time.
It really is. So our mission for this deep dive is based on a July 2026 paper published in Cell Reports Medicine. And we're going to explore how doctors are transitioning from just, looking at those scattered ingredients of a tumor to actually mapping its exact microscopic neighborhoods.
Right, to predict if a treatment will actually save a patient's life.
Yeah. And even if you aren't an oncologist, for you listening right now, you really need to hear this. It's a fundamental shift in how human biological data is processed.
And it's all being driven by a groundbreaking technology called spatial transcriptomics.
Which is such a mouthful.
It is. But it's essentially giving us a high-definition biological GPS.
I love that analogy. Now, to really appreciate this leap we're taking, we need to understand the human stakes here. Let's look at the patients in the clinical trial that provided the source material for this study.
Right. So the study focuses on 73 patients, and they were diagnosed with a condition called MIBC.
Which stands for muscle-invasive bladder cancer?
Exactly.
And just to clarify for everyone, muscle-invasive means the cancer hasn't just, formed on the surface lining of the bladder.
Right. It's dug its way deep into the actual muscle wall. It's aggressive.
So these patients, and the median age was 66, mostly male, about 85 to 95% across the groups, they're facing a really serious crossroad.
They are. The standard endgame for this stage of bladder cancer is something called a cystectomy.
Which is the complete surgical removal of the bladder.
And obviously, doctors want to avoid that if at all possible, or at the very least, they want to shrink the tumor drastically before they go in for surgery.
Right. So to do that, they want to hit the cancer with either standard chemotherapy or a combination immunotherapy.
But you can't just, flip a coin to decide which one.
No. You definitely don't want to pump toxic chemotherapy into a patient if their specific tumor isn't even going to respond to it. So how do doctors traditionally decide which drug to give?
Well, historically, and in the baseline of the specific trial, researchers take a biopsy of the tumor, and they run it through a machine called a nanostring encounter.
A nanostring encounter. Oh.
Yeah. It's a fantastic piece of technology for its era. And what it does is it looks for something called a tumor inflammatory signature, or TIS.
TIS, got it.
Basically, it counts the RNA molecules that are associated with inflammation.
So it's basically looking for signs of a fight.
Precisely. Think of the nanostring test like a thermal camera. Imagine flying a helicopter over our hypothetical city at night with a thermal camera.
Okay.
It scans the tumor and categorizes it into one of two buckets, hot or cold.
Like literal temperatures.
Well, inflammatory temperatures. If a tumor is hot, the thermal camera is lighting up. There's heavy inflammation, which tells the doctor that the patient's immune system is already at the scene, they've recognized the cancer, and they're trying to fight it.
Okay, that makes sense. And if the thermal camera reads cold,
then it's a ghost town. The immune system either hasn't recognized the threat at all, or it's been locked out of the tumor entirely.
Wow. Okay, so based on this thermal scan, the patients in the trial were randomized to receive treatments. But wait, let me just stop here for a second.
Sure.
Because this seems like a pretty logical system, right? Like, if a tumor is hot, we know the immune cells are there. If it's cold, we know they aren't.
Right.
So why do we need anything more complicated than this thermal camera to predict who is going to get better? It seems pretty cut and dry.
It really does seem like a perfect sorting system on paper. But, and this is big but, let's look at the actual clinical outcomes from the source data. Let's look at the group that received the immune checkpoint inhibitor, the immunotherapy.
Okay, let's hear the numbers.
So, yes, 38.5% of those patients had a complete response. The tumor vanished.
It's incredible.
And another 61.5% saw tumor downstaging, meaning the cancer shrank significantly.
So it's working. Those are great numbers.
They are. But look closely at the remaining data for that exact same hot group. A full 38.5% of patients had absolutely no response to the drug.
Wait, hold on. 38.5%.
Yeah.
So even if the thermal camera definitively proved the immune system was present at the tumor, almost 40% of those patients still didn't get better from the immunotherapy.
Exactly. And this is the absolute crux of the problem.
Wow.
The thermal camera tells you that people are in the city, but it doesn't tell you what they are doing.
Oh, I see.
It doesn't tell you if the firefighters are actually putting out the fire or if they are, trapped in gridlock traffic 10 blocks away.
Oh, that's such a good way to put it.
Yeah. The hot-cold binary label simply isn't accurate enough to predict who gets cured because mere presence does not equal engagement.
Right. Because knowing the ingredients are there isn't enough. We need to know what they're doing.
Yeah.
Okay, so let's do a really quick recap for you listening just to lock this in. So far, we've got 73 patients with aggressive bladder cancer. Doctors use a thermal camera, the nanostring, to see if the tumor is hot or cold.
Right.
But the big takeaway so far is that even when a tumor is hot, almost 40% of the time, the treatment still fails.
Exactly. That's the 40% blind spot.
Okay, so to solve this blind spot, the researchers initiated what they call a multi-omics characterization.
Right, multi-omics.
Basically, they attack the tumor's data from multiple angles. They did traditional DNA sequencing to find mutations, sure. But the real star of the show, the huge technological upgrade, is this thing called the 10x Xenium platform.
Yes, this is the spatial transcriptomics we mentioned at the very beginning. Xenium is it's a revolutionary piece of hardware.
How does it actually work, though?
So it allows researchers to identify exactly which genes are active in specific cells, but it does this while simultaneously preserving the physical tissue structure.
So they aren't grinding it up into rubble anymore.
Exactly. They even follow it up with a traditional H&E stain.
What's that?
It's a standard purple and pink tissue dye that pathologists have used for decades, but they do it right on the exact same glass slide as the Xenium scan.
Oh, wow.
Yeah. This allows them to visually see the cellular architecture perfectly overlaid with the gene expression data.
That is wild. And let me just ask what I think a lot of people might be wondering here.
Yeah.
If we already did a DNA spell check to find the mutations, and we already know what genes are active from the RNA, why do we care where the cell is physically sitting? I mean, isn't a cancer cell just a cancer cell, regardless of its zip code in the tissue?
And this is where we have to completely transition from chemistry to geography.
Okay.
Think of traditional bulk sequencing the way we used to do it, where we grind it up like a blender.
A blend?
Yeah. You take a piece of the tumor, drop it in the blender, and you make a molecular smoothie. You can test that smoothie and confidently say, I detect strawberry, I detect kiwi, I detect blueberry. You know exactly what ingredients are in the cup.
But you've destroyed the structure.
Right. The smoothie tells you the immune cells and the cancer cells are both in the cup. But spatial transcriptomics, the Xenium technology, it's like looking at a beautifully constructed fruit tart.
Oh, I like that.
Right. You don't just know there are strawberries and kiwis. You see exactly where the strawberry sits relative to the kiwi.
Okay, but biologically, why does seeing the fruit tart save lives while the smoothie fails? Why does that spatial relationship matter?
It all comes down to how our immune system actually operates on a microscopic level.
Yeah.
Let's take a T cell, for example.
The frontline assassins.
Exactly. For a T cell to kill a cancer cell, it must physically touch it. It has to.
It can't just shoot it from a distance.
No. The T cell has to bind to the tumor cell's membrane to inject toxic proteins like perforin and granzymes that literally blow the cancer cell apart from the inside.
So it's literally hand-to-hand combat.
Exactly. It is hand-to-hand combat. So if your immune cells are trapped far away from the cancer by a dense microscopic wall of scar tissue.
Oh, I see where this is going.
Right. Your tumor might look hot in the smoothie because the immune cells are technically inside the tissue sample.
The patient won't get better.
Exactly. The assassins can't physically reach their targets. The smoothie looks identical to a cured patient, but the spatial reality is a walled-off fortress.
That is absolutely mind-blowing. The geography of the cell literally dictates the survival of the patient.
It really does.
So, okay, once they had this Xenium technology to actually see the fruit tart, How did they make sense of it? I mean, a single tumor biopsy must contain, what, millions of cells?
It does, and it's incredibly chaotic. To make sense of it, the researchers had to establish borders. Yeah. They created a neighborhood matrix. They programmed their system to draw a strict 25 micrometer radius around every single cell in the sample.
Let's give that some context for the listener, because 25 micrometers is unfathomably small.
It is tiny.
Like a single human cell is usually somewhere between 10 and 20 micrometers across, right?
Right.
So 25 micrometer radius means you are looking at a central cell and its absolute most immediate next door neighbors.
Exactly.
You're looking at a microscopic bubble that is basically just 2 or 3 cells deep.
And within that tiny microscopic bubble, the technology identifies exactly who is living there.
Okay, so who are the major players they're looking for in this microscopic neighborhood?
They're tracking a highly specific cast of characters. They look for CD8 plus T cells, those are the frontline assassins we mentioned.
Right.
They look for CD4 plus T cells, which act more like generals, kind of directing the immune response.
Okay, assassins and generals.
Then they track B cells, which provide intelligence and antibodies. But, and this is where it gets really nuanced, they track macrophages.
Macrophages, those are essentially the cleanup crews, right? They swallow cellular debris.
They are, but macrophages can be a real double-edged sword in cancer.
How so?
Well, the researchers specifically had to differentiate between M1 macrophages, which are actively trying to attack the tumor, and M2 macrophages.
Wait, are M2 macrophages bad?
Very. Cancer is incredibly deceptive. It can actually hijack M2 macrophages and totally brainwash them.
Brainwash them to do what?
To build new blood vessels to feed the tumor instead of fighting it.
Wow. So it's literally turning the cleanup crew into enemy contractors.
That is exactly what it's doing. And they also looked for TREGs, or regulatory T cells.
What do those do?
They normally act as peacekeepers. They stop your immune system from overreacting and attacking your own healthy organs. But cancer can exploit these peacekeepers to suppress the assassins.
Of course it can, sneaky.
Very. And finally, they tracked fibroblasts, which build structural scaffolding, and endothelial cells, which form the blood vessels.
Okay, so when you lay out all these different cells and their roles, it really brings us right back to that city analogy from the intro.
It perfectly maps to it. Drawing that 25 micrometer circle is exactly like city zoning. You take this massive, chaotic tumor, drop a circle the size of a city block, and you just count the buildings.
I love this. So if the block is packed with organ cells and tumor cells, it's a residential zone.
Right, or what they call epithelial rich in the paper.
Okay. And if it's full of fibroblasts and structural scaffolding, it's an industrial zone.
Exactly. And if it's packed with macrophages and T cells, well, that's a police station.
But, okay, doing this for millions of cells, you can't manually zone a city that large. I mean, it would take a lifetime.
Oh, absolutely. Which is why they brought in some really heavy-duty data science. They used algorithms, specifically one called k-means clustering, to organize all this chaos.
Oh, k-means clustering. That is a fascinating tool. For those who aren't familiar, imagine you have a giant map covered in thousands of scattered pins. K-means is this algorithm where you drop a set number of central hubs onto the map, and the computer calculates the distance from every single pin to every single hub.
It's all about proximity.
Exactly. Every pin gets assigned to whichever hub it is mathematically closest to. So it groups massive amounts of data by proximity and similarity.
Right. It groups similar microscopic neighborhoods together. But, you know, the main challenge with k-means is that you have to tell the computer how many hubs to drop in the first place.
Right, because the computer doesn't inherently know how many zones there should be.
Exactly. Like, should we divide the tumor into 4 zones, 10 zones?
So how did they figure that out?
They use something called the elbow method.
The elbow method?
Yeah, you basically plot the variance of the data on a line graph. As you add more clusters, more hubs, the error rate drops sharply, which creates this steep downward line.
Okay, I'm visualizing a graph going down.
But eventually, adding more clusters doesn't really give you any new, useful information, and the line just sort of levels off horizontally.
Ah, so the point on the graph where the line bends, where it goes from dropping steeply to going flat, that looks like an elbow.
Exactly. That elbow tells you the optimal mathematical number of groups.
That is so clever.
It is. And using the elbow method, along with some quality control algorithms like Seurat, to filter the single-cell data, and DB-scan to identify dense clusters.
That is a lot of computational firepower.
It is a massive computational effort, but it revealed something incredible. The algorithms grouped all these millions of microscopic city blocks into exactly 7 distinct communities.
7 distinct microscopic neighborhoods within the bladder cancer.
Yeah.
So let's do another quick recap for everyone listening. We've gone from looking at a blended up tumor smoothie to drawing tiny 25 micrometer bubbles around every cell, identifying the assassins, the brainwash cleanup crews, the structural scaffolding, and then using k-means clustering to organize all that chaos into 7 specific communities.
You got it.
Okay, so what do these 7 communities actually look like?
Well, looking at the extremes is probably the most illuminating part of the paper. For instance, the algorithm identified Community 6.
Okay, what's Community 6?
This zone was highly epithelial-rich, meaning it was packed tightly with the tumor and organ cells, but it was completely macrophage-poor.
Wait, meaning there were no immune cleanup crews allowed inside?
None. It was a totally walled-off residential zone just for the cancer.
That's terrifying, but amazing that we can see it, and I imagine they found the inverse as well.
Yes. Community 2, for example, was the exact opposite. Heavy immune presence, very low tumor density.
Wow.
So by defining these 7 communities, they successfully turned abstract, invisible genetic data into a visual, quantifiable architectural blueprint.
It's no longer just a list of mutations in a spreadsheet.
Exactly. It is a high-definition map of a microscopic war zone.
Okay, this is where we really need to connect the dots for the listener.
Let's do it.
Because we have this incredible data science. We have mapped the 25 micrometer neighborhoods. We have 7 communities. But how does knowing that a tumor has 7 distinct neighborhoods actually help that 66-year-old patient waiting in the hospital for a cystectomy?
That's the real question.
Right. Like, how does a zoning map actually save a life?
Well, that is the ultimate goal of this entire Cell Reports medicine paper, clinical translation.
Translating the science to the clinic.
Exactly. We have the map. Now we need to use it to predict the cure. To do this, the researchers didn't just map where the cells lived. They mapped the highly specific communications happening between the cells.
Ah, they tracked the immune checkpoints.
This is vital. Immune checkpoints are essentially safety switches on your immune cells.
Okay.
The researchers used the Xenium map to track exactly where a protein called CD86 interacts with CTLA4.
Well, I've heard of that.
Or even more famously, they tracked where a protein called PDL1 interacts with PD1.
Yes. The PD1 and PDL1 interaction is so critical to understand here because this is the exact mechanism that a lot of modern immunotherapies target. And for you listening, I think the best way to visualize the PD1 receptor on an immune T cell is to think of it like a barcode scanner.
That is a great analogy.
Right. So the T cell is wandering through the tissue, and it uses its PD1 scanner to check the IDs of the cells it bumps into, making sure they are healthy host cells and not threats.
Just checking them at the door.
Exactly.
Right.
But cancer cells are incredibly devious. They mutate to cover themselves in PDL1 proteins.
Which act as a fake ID.
Yes, a fake ID. So when the T cell's barcode scanner reads the tumor's fake ID, the scanner registers friendly. The T cell is tricked into basically going to sleep instead of attacking the cancer.
It's completely disarmed.
Right. And immunotherapy drugs work by blocking that fake ID, allowing the immune system to wake up and see the cancer for what it actually is.
Precisely. Now remember our thermal camera from the beginning?
The smoothie approach.
Right, the smoothie approach. In the past, doctors just looked to see if the fake IDs were present anywhere in the blended cup.
Oh, but they didn't know if the scanner was actually touching them.
Exactly. But with spatial transcriptomics, researchers looked inside those 25 micrometer neighborhoods to see exactly where the scanner was physically touching the fake ID.
Wow.
They mapped the exact geographical points of immune suppression.
So when an oncologist looks at this new Xenium map, what are they actually looking for to make the call on treatment?
They are looking for the spatial relationship. They want to see if the police, the T cells, are actively penetrating the criminal tumor communities and getting suppressed there.
Because if they are, the immunotherapy will wake them up and they're already in position to fight.
Exactly. Or they want to see if those T cells are walled off in a fibroblast industrial zone, literally miles away from the cancer microscopically.
Because if they're miles away, waking them up won't do any good.
Right.
So does seeing that geography actually tell the doctor if the patient will be in that lucky 38.5% who get a complete response?
Yes. That is the holy grail they are building here. By quantifying this spatial urban warfare, doctors can predict drug success before they ever administer toxic treatments.
That is incredible.
If the map shows a walled-off fortress where the T cells can't physically reach the tumor, you don't give the patient immunotherapy. It simply won't work, no matter how many immune cells are hovering on the outside.
You save them the toxicity?
Exactly. You save them the toxicity and you immediately try a different strategy, like chemotherapy or just going straight to early surgery.
It really takes a biomarker from being just a simple binary hot or cold test and turns it into this dynamic, predictive engine.
It really does.
It's the ultimate promise of personalized medicine. You aren't just treating bladder cancer generically. You're treating the specific architectural layout of this specific patient's bladder cancer.
It is a monumental shift in how we approach the disease. I mean, we are really moving from genetics to geography.
What an incredible journey through the data today. I mean, we started in the murky waters of the thermal camera, where doctors were blending up tumors and wondering why nearly 40% of patients with hot immune signatures weren't responding to treatment.
And we found the answer by keeping the tissue intact. We moved from the smoothie to the fruit tart using the 10x Xenium platform.
We watched algorithms draw 25 micrometer neighborhoods, creating 7 distinct microscopic communities. And we saw how mapping the exact urban warfare, literally tracking the physical touch of a fake ID, gives doctors a crystal ball to predict which treatments will actually work.
The overarching lesson here is that in human biology, structure is function.
Structure is function.
Location isn't just a detail, it is the determining factor in whether a drug succeeds or fails.
Well said. So for you listening, the next time you hear the word biopsy or biomarker in the news, remember this deep dive. A tumor isn't just a lump of bad cells. It is a complex, geographically diverse city.
It really is.
And to close this out, I want to leave you, our listener, with a provocative thought to sort of mull over on your own.
Oh, all right.
If spatial transcriptomics can map the microscopic battlegrounds of bladder cancer to predict exactly which drugs will work, what happens when we turn this spatial technology on the brain?
Wow, that's a profound thought.
Right. Could diseases like Alzheimer's or Parkinson's, which have honestly baffled neurology for decades, could they just be complex neighborhood disputes that we simply haven't had the right maps to see yet?
It's an incredible question. I mean, when the old diagnostic tools fail, you don't give up on the patient.
No.
You invent a better way to see the city.
Exactly.
Yeah.
You invent a better way to see the city. The diagnostic muddy waters are finally clearing up. Thanks for diving deep with us today.